Hi guys, welcome to my YouTube channel. This is Arun. In this video we are going to see about the cattle infecting bacteria Mycobacterium bovis. Mycobacterium bovis is also called as bovine tuberculosis. First, general characteristic. Mycobacterium bovis is a gram-positive acid-fast bacilli. It is rod-shaped bacteria. It belongs to the family Mycobacteriaceae. Motility in non-motile in nature and capsule and endospores is absent. The mode of respiration is aerobic respiration and optimum temperature is 25 to 37 degrees Celsius and optimum pH is 6.4 to 7.0. Habitat is Mycobacterium bovis causes tuberculosis in cattle and in humans. Most are found in soil and water in a free living form of in diseased tissue of animals. Discovery in 1908, French scientists Albert Calmet and Camille Guerin chose a Mycobacterium bovis strain for their work on tuberculosis vaccine. Pathogenicity of Mycobacterium bovis, disease and transmission. Mycobacterium bovis causes bovine tuberculosis affecting cattle primarily. It can also infect humans and other animals. Transmission occurs through inhalation of respiratory droplets, consumption of contaminated milk or meat, and direct contact with infected animals. Control measures include testing and pasteurization of dairy products. Incubation period of Mycobacterium bovis takes months to development of bacteria in the host cell, dormant for years and reactivating during period stress or old age, 4 to 12 weeks. Virulence factors of Mycobacterium bovis: cell wall, mycolic acid, trialase 6,6-dimycol, CS86 and CFP10 lipoarabinamide. Pathogenicity of Mycobacterium bovis. Mycobacterium bovis, a bacterium related to one causing tuberculosis, exhibits pathogenicity by infecting various animals, particularly cattle. This pathogen can be transmitted to humans through consumption of contaminated dairy products. Once inside the host, Mycobacterium bovis evades the immune system, forming granulomas in tissues. The persistent infection contributes to the bacteria's ability to cause disease, impacting both animal and human health. Clinical disease of Mycobacterium bovis. Mycobacterium bovis can cause clinical disease in various animal species, particularly in cattle, and it can also lead to zoonotic infection in humans. The clinical manifestation of Mycobacterium bovis infection can vary depending on the species and its affected organs. Here are key aspects of clinical disease caused by Mycobacterium bovis. Next, in cattle, Mycobacterium bovis is a major causative agent of bovine tuberculosis. Infected cattle may exhibit chronic cough, weight loss, weakness, and respiratory distress. Disseminated infection: the bacteria can spread to different organs, leading to lesions in the lungs, lymph nodes, liver, and other tissues. The dissemination cause result in systemic disease. This image shows the lung infection in cattle. In humans, zoonotic transmission, pulmonary tuberculosis. Human infection with Mycobacterium bovis can lead to pulmonary tuberculosis, persisting with symptoms such as cough, chest pain, weight loss, and fatigue. It resembles tuberculosis caused by Mycobacterium bovis. Extrapulmonary infection. In rare cases, Mycobacterium bovis can cause extrapulmonary infection affecting organs outside the lungs. This can include infection in bones, joints, and central nervous system. This image shows the lung infection in human beings. Laboratory diagnosis of Mycobacterium bovis. Diagnosis of Mycobacterium bovis involves laboratory methods. Common approaches include: first one is microscopy. Acid-fast staining of clinical specimens like sputum or tissues helps in identify of Mycobacterium, including Mycobacterium bovis under a microscope. Second one is culture. Isolation of Mycobacterium from clinical specimens and specialized media such as Lowenstein Jensen is a key step of Mycobacterium bovis forms distinctive colonies. Third one is PCR, polymerase chain reaction. Molecular techniques like PCR amplify specific DNA sequence of Mycobacterium bovis, aiding in rapid and accurate identification. Fourth one is immunological test. Serological test detects specific antibodies against Mycobacterium bovis antigens. providing additional diagnostic information. Fifth one is radiology. Imaging studies like chest x-rays may reveal characteristic patterns associated with Mycobacterium bovis infection, assisting in diagnosis. Next, colony morphology and culture medium of Mycobacterium bovis. Mycobacterium bovis colonies exhibit distinctive features. First one is appearances: rough, irregular, and dry colonies. Color: typically cream to yellowish or buff. Growth rate: slow growth with colonies taking several weeks to develop. Mycobacterium bovis is commonly cultured and specialized media designed to support the growth of mycobacteria, and such as medium is Lowenstein Jensen medium contains ingredients like egg or serum, malachite green, and glycerol supporting the growth of mycobacteria. Middlebrook 7H10 and 7H11 agar selective and enriched media designed for cultivation of mycobacteria. Liquid media, example Middlebrook 7H9 broth, provides a more rapid growth environment compared to solid media. Treatment: antibiotic therapy including drugs like isoniazid, rifampicin, ethambutol, pyrazinamide, and streptomycin is commonly used. Multi-drug requirements are employed to prevent antibiotic resistance. Patients are closely monitored for treatment response and potential side effects. Finally, prevention of Mycobacterium bovis. Vaccination in animals. Some regions use Bacillus calmette-guerin vaccine to reduce Mycobacterium bovis infection in cattle. Pasteurization. Proper pasteurization of dairy products is essential to prevent human transmission. Animal health measures. Implementing biosecurity measures, testing and culling infected animals, and controlling animal movement help to prevent spread. Public education, promoting awareness about this associated with unpasteurized dairy and encouraging hygiene practice reduce the risk of human infections. Thank you.